So in this lecture, we're going to talk about the physical and chemical control of microbes. If you look at the average supermarket aisle or Walmart aisle, you're going to see a whole bunch of different products that have been developed in order to destroy microbes, or at least control the growth of microbes. Some might say that we are truly obsessed with finding ways to control microbial growth. And that's what we're going to talk about today. And before we get into specifics, I want to kind of give you an idea of the relative resistance of microbes. Of course, you already know that by far the most resistant of all microbes are bacterial endospores. Bacterial endospores are much hardier than any other life form on the planet. They are without a doubt the hardest thing to kill. They're about 18 times harder to destroy than the vegetative form of those organisms. And in fact, their destruction is the goal for any sterilizing process. So something like the canning industry, their goal is to destroy bacterial endospores. So without a doubt, they have this really, really high resistance. There are also some that are sort of in a moderate range. A couple of bacteria that tend to be more resistant to chemicals than other bacteria: Pseudomonas, Mycobacterium, and Staphylococcus aureus. Pseudomonas is notorious for his ability to grow in lots of different environments. In fact, when we talk about him with the pathogens, I'm going to talk about how hardy he is, that he's even found to be growing in things like soap dishes, and he'll grow in the containers of disinfectant. Just a really, really hardy bacteria. Mycobacterium tuberculosis. Mycobacterium, because of its mycolic acids, tends to be more resistant than the average bacteria. Staphylococcus aureus tends to be more resistant than the average bacteria because it turns out that staph is slightly acidophilic and can also grow in cold temperatures and can also handle salt, and so he is very hardy in how many different environments he can grow in. If we compare something like protozoans, the protozoan cyst is more difficult to kill than the protozoan trophozoite. The cyst is a little bit hardier. If we compare viruses, the naked viruses tend to be a little bit more resistant than the enveloped viruses. The naked viruses, because they don't have that membrane around them, and the membrane creates a target that you can destroy those viruses with. So least resistant here. Vegetative bacterial cells are pretty easy to kill. Protozoan trophozoites are pretty easy to kill. Envelope viruses, because they have a membrane we can dissolve, are pretty easy to kill. And then fungi really just don't have any chance whatsoever. Now, some terms that deal with this area that get thrown around incorrectly all the time. First here, the term sterilization. Sterilization is a process that destroys all viable microbes. It destroys everything, including viruses. It even gets endospores. So you could sterilize equipment. Obviously, that you cannot do that to your patient's skin. So sterilization is only an appropriate term for what you're doing to inanimate objects. In fact, all of them that I have in purple here are terms that deal with inanimate objects. The ones in orange deal with body parts. All right, so sterilization destroys everything, including endospores. Disinfection is a process that destroys vegetative pathogens but doesn't kill endospores. That's a very common one that will disinfect. We're not actually killing spores, but we're killing everything else. Sanitization is a cleansing technique that mechanically removes microbes. So if you're like scrubbing, right, scrubbing a cutting board or a work surface, then you're mechanically removing microbes. You're sanitizing. Now you can mix two terms together. So if you were scrubbing with an antimicrobial soap, you would be doing both sanitization and disinfection at the same time, where you were using the soap that was disinfecting while you were sanitizing by scrubbing. Again, sterilization, disinfection, and sanitization are all terms that are appropriate on inanimate objects. The term degermation. Degermation is something that reduces the number of microbes on the skin. So for example, when you wash your hands, you're degerming them. Antisepsis. Antisepsis is when you use a chemical agent to destroy or inhibit vegetative pathogens. And that's a term that has to do with body parts as well. So degermation, antisepsis has to do with human bodies. Right? So for example, if you were washing your hands with antimicrobial soap, you would be doing both degermation and antisepsis. So you can mix terms together. Microbial death. Just like when we talked about growth, microbial death is a little bit different than it is for macroscopic organisms. Microbial death, the term death, is difficult to describe. We're not talking about one particular cell, but instead we're thinking about the culture as a whole. So how do we define death? Death is defined as the permanent loss of reproductive capability, even under optimum growth conditions. So if that microbe is truly dead, if you were to put it into optimum growth conditions, it still would not grow. Right? That's what death means, is permanent loss of reproductive capability, no matter what the conditions are. Now, as you can imagine, in a culture, you have lots and lots of organisms, and they don't just die at the same time. It's not like all of the cells go, okay, 1, 2, 3, die. In fact, there is a death rate. And you can see that here, where as you expose it to whatever your agent is, right, over time, the number of live cells drops. So there is a death curve, a death rate, just like there would be a growth rate. Now, what I want to talk about is some things, some factors that affect death rate, right? Some of the factors that can affect death rate. And the first one here is exposure time. How long is the culture exposed to the agent? And it makes sense that in general, the longer the culture is exposed to whatever your agent is, whatever the chemical or whatever it is, right? The longer your culture is exposed to it, the faster they die. Right? That's what you're seeing here. The type of organism affects death rate. Some organisms are hardier than others. If you have only vegetative cells, they'll die relatively quickly. If you have spores, they'll die much more slowly. Right? So the type of organism matters. The microbial load matters. In other words, how many microbes are in that culture. If you have a culture that's very turbid with lots of microbes, it will take longer to kill them all than if you have a culture with low load. Okay. What's called the agent's action matters. There are some agents that are what we call microbial-cidal. Microbial-cidal means it truly kills the microbe. That's different than the term microbiostatic. Microbiostatic means it stops the growth but doesn't kill the microbe. Right? So that makes a difference. Is your agent microbiostatic or is your agent microbiocidal? Cidal means permanent loss of reproductive function. But microbiostatic means that if you were to return it to a good condition, then that culture would begin to grow again. Those are two different things. Sometimes you see more specific terms, by the way, where people will call something bactericidal or fungicidal or virucidal. They're being more specific as to what exactly it destroys. All right. Next here. Let's talk about environment. Right? The environment that your culture is in can drastically affect how successful your killing agent is. So some chemicals require a certain temperature range or pH range to function well. Right? And if it's too hot or too cold or too acidic or too basic, it's not as effective at killing your culture. Concentration. The concentration of whatever your agent is, whatever your chemical is. And in general, as you can imagine, the higher the concentration of the chemical, the faster it kills. Now, that's true almost always, that the higher the concentration, the faster it kills. There is one notable exception, however. Alcohol, ethanol. Right? Ethanol, it turns out that ethanol must have water to kill, and so, in fact, 100% ethanol is not as effective as 90%. So ethanol is the one and only exception to that rule. Generally, the higher the concentration, the more effective, but that's not true for ethanol. All right, organic matter. If your culture is in an environment with lots of organic matter, that can slow down the death of the culture. Sort of bogs the chemical down with all of those organic molecules there. Let's do an example. So if we were to take 1 milliliter of E. coli and pour it onto a desktop, and then we took the same amount of E. coli and poured it onto a pool of vomit. Right? Vomit has a lot of organic matter in it. Well, what's going to happen is it will require much more of your chemical to kill off the E. coli that's on the vomit than it will to kill the E. coli that's just there on the countertop. And the reason is because of all of that organic matter. It literally sort of bogs the chemical down and slows down the process. It requires significantly more. In fact, when you have a spill, if you have a spill with a lot of microbes, I'll oftentimes tell you to spray it down, put your paper towels on top of it, let it soak, wipe it up, spray the area again, wipe that up, because I want to make sure we've gotten through all of the possible organic matter that's there. And of course, this becomes really important in medicine. If you think about what's happening in medicine, is you're pretty much always cleaning up something that's high organic matter. So blood, feces, saliva, vomit, and all that stuff, right? That's all going to be high organic matter, and you have to consider that when you're using a chemical to kill off possible pathogens. All right, so when we do kill pathogens, some common antimicrobial targets. So parts of the microbe that are commonly targeted in order to destroy them. The cell wall. Either we can stop the synthesis of the cell wall or we can destroy the cell wall. Either way, the cell will lyse and die, and that's deadly, absolutely. The cell membrane. If you dissolve the lipid bilayer, that's going to kill the cell. Stopping synthetic processes. If we stop DNA replication or stop transcription or stop translation or stop metabolism, we're going to destroy the cell. Proteins, and I mean specifically denaturing proteins. If you do something to denature the proteins of the cell, remember that proteins make up all of the structure and function of the cell. So if you disrupt those proteins by denaturing them, then the cell will be killed. All right. In this lecture, we're going to go through several different types of control mechanisms here. We're going to talk about some physical agents first, working our way through this side, and then we're going to talk about some mechanical agents, and then we'll get into the chemicals last. We're going to kind of work our way through these different agents. For each one, you should know what it kills and how it kills. Those are kind of the major take-home points for each of these individual agents. All right, let's talk about in red here these physical agents of microbial control, right? And this is heat, cold, desiccation, which is also called dehydration, and radiation, right? These are all methods for physically controlling the growth of microbes. We're going to work our way through each of them one at a time. Starting out with heat. I want to talk first about moist heat. Moist heat is using hot water or steam, and you're raising the temperature above the max growth temp. This is microbicidal. It does kill off microbes. It is a microbicidal technique to raise it above their maximum growth temperature. Now, how does it do that? How does it kill off the microbe? It does that by denaturing its proteins. Remember, proteins are only effective when they are shaped correctly. If you either change the pH dramatically or increase the temperature, they will change their shape and they're no longer active. And this is exactly what happens when you fry an egg. Put the egg into the frying pan, and the egg white, which starts out as that clear gloppy material, eventually becomes white, opaque, and rubbery. And what's happening is all of the normal egg white proteins, when they get hot enough, are denaturing and changing their shape. And just like you can't unfry the egg, you can't put those proteins back together again either. So this is microbicidal. You can even get hot enough to kill spores if you use an autoclave. So you can achieve sterilization using an autoclave. An autoclave is basically a great big pressure cooker, and this is how Mary gets rid of all of our waste in the lab. Here's how it works. There's this chamber here where she puts all of our waste in, like all of your tubes after you're done taking your labels off. She puts them into this chamber, cranks the door closed. There's this fancy locking mechanism. And then turns it on, and it starts to bring steam into the chamber, and it will hold the pressure really high to force the temperature up hotter and hotter and hotter. And how hot do you have to get in order to truly kill off even spores? You have to get to 121 degrees Celsius, and you do that by getting to 15 psi pressure for about 15 minutes. Now the time depends on how much stuff you have in the chamber, but about 15 to 30 minutes is pretty standard. So in order to achieve sterilization, you can use moist heat, but you got to use an autoclave that gets up to 121 degrees Celsius at 15 psi, and it kills by denaturing proteins. Now there are other types of moist heat. Pasteurization is a process that was developed by, yep, you guessed it, Louis Pasteur. Louis Pasteur came up with this technique for achieving disinfection but not sterilization. This is a process that you use on a liquid. Now the goal here of pasteurization is to kill off vegetative pathogens but not completely destroy your liquid's flavor and nutrient value. For example, if we were to take your carton of milk and put it through the autoclave, now we could completely sterilize that milk so that there was no pathogens left in it, but if you think about what you're likely to get out of the autoclave, you'd get like a big glob of milk goo because everything would not be in its normal shape and certainly not its normal flavor. So pasteurization is trying to find a balancing act. The goal is to get hot enough that you can kill off pathogens, but not so hot that you denature the flavor and the nutrients of whatever your liquid is. That's what pasteurization is about. So how hot do they get it? The old school way of pasteurizing was 30 minutes at 66 degrees Celsius. They have newer techniques that use 15 seconds at 71.6 degrees Celsius and achieve about the same amount of killing. Excuse me. So again, this is a term that applies to liquids, and it's there just to find a way to try and balance this out. Now, if you think back, what kind of microbes are usually pathogens? Are they acidophiles? Are they neutrophils? Mostly mesophiles. Most of the human pathogens are mesophiles, meaning they grow at a moderate temperature. So if you get hot enough, if you get up to 66 degrees Celsius or 71.6 degrees Celsius, that's hot enough to denature them and kill them. So this technique does kill pathogens that are mostly mesophiles without actually destroying the milk or whatever other liquid it is. Dry heat. Now dry heat is a horse of a totally different color. Dry heat is not as effective as steam heat. It requires much higher temperatures than moist heat. You can sterilize if you get hot enough, but it does take quite a bit hotter to sterilize using dry heat than it does using steam. So for example, dry ovens, if you want to use that as a sterilization, you could do that, but it takes 150 to 180 degrees Celsius to get hot enough to denature proteins. Labs who don't have autoclaves that are forced to use dry ovens generally will load up the dry oven before they leave for the evening, turn it on, and leave it all night because it takes an extended time period to get that hot and maintain it hot enough, long enough to kill everything. Now, the other possibility is incineration. Incineration is what you do when you have a loop full of microbes and you put them through the Bunsen burner. You're doing incineration. Your Bunsen burners only get up somewhere around 800 degrees, but it could be much, much hotter if you think about like commercial incinerators. In incineration, they are completely oxidizing and combusting the cells, returning the cells to carbon dioxide and water vapor. Of course, that sterilizes as well. So you can sterilize using dry heat. Now, cold temperatures, on the other hand. Cold temperatures are microbiostatic. Very few microbes are killed off by cold temperature. Cold temperature is a microbiostatic phenomenon. It slows the growth of microbes. Cold temperatures we think of as being in the refrigerator or freezing, which would be underneath lower than 0 degrees Celsius. Cold temperature is usually used to preserve food, media, and cultures, but it's not going to kill them off. As soon as you return them to warm temperatures, the microbes will grow just fine. In fact, sometimes you guys will inoculate your culture tubes, and I'll put them in the fridge until Monday or Tuesday, and then I'll put them into the incubator, and they're just fine. Cold temperatures inhibit growth, but does not kill microbes. Desiccation is very similar. Desiccation, better known as dehydration, leads to metabolic inhibition. Water is absolutely essential for metabolism to go on. So if there's no water, then no metabolism is happening, and therefore the cell is inactive. Right? Now, this is not microbiocidal. This is microbiostatic also. As soon as water is returned to the microbes, they will be just fine. For example, if I wanted to send a sample of my E. coli culture to my friend who's a researcher in New York City, what I would do is take that E. coli sample, completely desiccate it. You can do this with this machine that will pull out all of the liquid, and the bacteria will become like a little powder in the bottom of the tube. I send that tube through the mail. My friend gets the tube in New York, she adds in some fresh broth, and there you go, they're fine. So dehydration is microbiostatic. It inhibits growth, but as soon as the liquid is returned, those microbes will be fine. Another physical control mechanism here, this is radiation. Radiation, there's really two different kinds of radiation, ionizing and non-ionizing. Let's look at the differences here. Ionizing radiation causes breaks in the DNA. That's what you're seeing here. The kind of damage that it causes is these breaks in the DNA. And some examples here, excuse me, I don't know why I keep yawning. Some examples here of ionizing radiation: gamma rays, X-rays, cathode rays. Those are all ionizing radiation that cause breaks in the double-stranded DNA. You can sterilize medical supplies or food products using ionizing radiation. It is effective, especially because ionizing radiation can go through barriers. It can transmit through, it does penetrate, that's the term I'm looking for. It does penetrate through various different things. So one thing they use ionizing radiation for now is when you buy like hamburger meat. You buy hamburger meat, it's in this great big glob of meat on top of the little styrofoam dish. Well, some places what they're doing is those little styrofoam dishes are moving under a gamma or a cathode ray. And the gamma or the cathode ray goes through and it will penetrate through that big clump of meat and destroy all of the DNA that it encounters. So it can penetrate through barriers, destroying DNA as it goes. So it will completely kill off any microbes that happen to be in there. It will also destroy the cow's DNA, but it doesn't matter. That's not what you taste anyway. Now, non-ionizing radiation. Non-ionizing radiation has very little penetrating power. In fact, it doesn't penetrate through very much. UV light is the most common example of non-ionizing radiation. UV light, you know, causes mutations in the DNA. If you remember from lab, what kind of mutations does UV light cause? UV light causes thymine dimers. It causes neighboring thymines to bind to one another, and you get that little bulge in the DNA. And that stops transcription, it stops replication, all of those processes. So UV light has very little penetrating power. It cannot penetrate through a barrier. In fact, it won't go through your sunglasses. You don't get sunburned through your t-shirt because all of those things block UV light. So what can UV light be used for? It is used to sterilize air, water, or solid surfaces. There are some hospitals who recycle the air through a UV light, so it goes under the UV light as air is circulating to kill off mold spores or bacteria that could be floating in the air. Some restaurants and now some surgical suites are equipped with UV lights. So the last person out at the end of the day turns on the UV light, and the UV light will shine down and sterilize surfaces, cutting boards or surfaces of the surgical suite in order to sterilize those solid surfaces. Now, it would never be effective to use on something like that big chunk of hamburger meat because the UV light won't penetrate, but it will work just fine on the surface or on air. All right. So we've talked about these physical agents and how they kill and what they kill. I now want to move over to this side and talk about the mechanical agents and how they can get rid of microbes. And really we're talking mainly about filtration and then washing and scrubbing. So methods of mechanical removal here, we can filter out microbes, or we could wash and scrub to mechanically remove microbes. Let's look at filtration first. Filtration is the physical removal of microbes by passing a gas or a liquid through a filter. And this works pretty much the same way as a colander does when you make spaghetti. So for example, here you have a little blow up of what's happening with our filter. Right? On this side, you have your liquid, and the liquid has the microbes in it. Here you have your filter. This is a filter paper. Now the pores of the filter paper are so small that the liquid can pass through, but the microbes cannot pass through. It's just like if you pour your pasta water through a colander, the colander catches the spaghetti and lets the fluid go through, right? That's what's going on here. So physical removal of microbes by passing a gas or a liquid through a filter. Now the question is, how well does this process work? Can you actually achieve sterilization? Now in this case, to achieve sterilization, you want to know just how small are those holes. The smallest of all microbes are the viruses. So the question is, are the holes small enough to keep viruses out? And the answer is yes. You actually can buy filters that have holes so small that they will block viruses. So it is possible to achieve sterilization by running something through a filter, because sometimes you can buy filter paper where the holes are so small viruses can't pass. So this is a great way to sterilize something that's heat sensitive. You can sterilize heat sensitive liquids or air in a hospital. Many times air in a hospital ward will be circulated through one of these filters to pull something out of the air if they don't have a UV light. You can use this for all sorts of different cases like that, where something can't be autoclaved, but it can go through a filter. For example, you don't want to autoclave something like IV fluids because you would break down some of the things that are in the IV fluids, but you can send it through a filter to make sure that it's sterile. Here's a picture of a piece of filter paper. You can actually see that this filter paper, you can see the individual pores for the liquid to go through. Here you can see a bacteria was caught on that filter paper. If you look at that bacteria, it's one we've seen before. That's Staphylococcus, round cells in irregular clusters. That's been caught there by that filter. In fact, you likely have a filter on some product at home, most commonly vacuums. Many vacuums now have what they call a HEPA filter. HEPA stands for High Efficiency Particulate Air Filter. HEPA is High Efficiency Particulate Air Filter. They have them on all kinds of stuff now, and they're meant to catch things as it passes through that filter and let the air pass through, but keep those molecules from passing through. All right. Washing and scrubbing, another mechanical removal technique here. I've always loved this picture. I just think it's such a scream, this late 60s hairdo. But anyway, washing and scrubbing hair. Sanitization is the mechanical removal of microbes from an inanimate object. So you can sanitize a benchtop. You can sanitize a cutting board. Right? You can also do disinfection. Disinfection is when you use a chemical to kill them. So you can both sanitize and disinfect your cutting board or your benchtop to both kill and remove all at the same time. The terms for the same sort of activity on human body parts would be degermation and antisepsis. Degermation reduces the number of microbes on the skin, and antisepsis is using a chemical agent to kill them. So if you think about something like a surgical scrub, doing both degermation and antisepsis. The act of scrubbing is the degermation, and the antimicrobial soap is the antisepsis to both destroy and remove microbes at the same time. All right, so again, if we look at control mechanisms, we talked about physical agents. We talked about mechanical agents. I now want to talk about some chemicals that are commonly used to kill microbes. Now, there are a whole bunch of different chemicals that are currently being used to kill microbes. No doubt, hundreds and hundreds of them. But really, most of them, almost all of them, fall into one of 9 different groups. So we're going to talk about these 9 different chemicals and how they are used to kill microbes. And the idea of using chemicals to kill microbes has been around for a very long time. In the 1800s, lye and iodine solutions were used in order to keep microbes from being able to grow. And in fact, there are over 10,000 different antimicrobial chemical agents that are currently available to keep microbes from being able to grow. Let's go through them one at a time. So the halogens here. The halogens, if you remember from the periodic table, the halogens are group 7. I'm counting in my head. Yes, group 7 of the periodic table are the halogens. That includes fluorine, bromide, chloride, iodine. Those are all the halogens. The 2 that are most commonly used as antimicrobials are chlorine and iodine. Let's talk about chlorine first. Chlorine is one of the most common, probably the most common chemical agent that's used to kill microbes. In fact, 1/3 of all marketed antimicrobial agents have chlorine as their active agent. It's been used for over 200 years, so we've known for a very long time that chlorine is an effective killer. Hypochlorite, right, which is bleach, that has chlorine in it, and chlorine is actually the active agent in bleach that kills off microbes. Now, how does it do that? Bleach kills, or all chlorine kills, by denaturing proteins. It can be even sporicidal if you are in high enough doses. Now remember, for each of the chemicals, you're going to want to know what it kills and how it kills. So chlorine denatures proteins. It can be sporicidal if the dosage is high enough. We use chlorine for all sorts of different things. It's the active killer in bleach. It's also used to disinfect water. Remember that chlorine is often added to water supplies, swimming pools, things like that, in order to kill off microbes. It turns out that you only need a very, very low level of chlorine, about 0.16 parts per million, in order to kill off microbes. So the idea is you can use such small doses that you can achieve killing without changing the flavor of the water. Now, some of you will tell me that the water tastes like chlorine, but it's relatively low, and most people can't taste the chlorine. That's why they like it so much. Now, the downside to chlorine is that it is sensitive to sunlight. It does break down over time, especially with exposure to sunlight. This is why bleach comes in an opaque container. Because sunlight does break down chlorine and its activity. This is why you have to keep adding more chlorine to your swimming pool, because the sun does break down the chlorine that you added last week. I will never forget this. One time I was in my lab for grad school, and when I had first started at grad school, I had made this chlorine solution to use as a disinfectant on my bench top, and my major professor came through like a couple years later. He comes through and he asked me how long that chlorine solution's been sitting there, and I said, oh, I think I made that when I first came. It's probably 2 years old. And he said, don't you know that chlorine is broken down if you let it sit too long? He unscrewed the top of the container and drank it. Now, definitely that's not a good idea, but it really sent home the message to me that chlorine is broken down over time, especially with exposure to light. Probably that'll help you remember it too. Now, a close relative of chlorine is iodine. Iodine also kills by denaturing proteins. It can also be sporicidal. It is a very useful agent for controlling microbes. And actually, iodine is not as sensitive to the sun as chlorine is. So iodine, in some ways, is better. So the question is, why aren't we using iodine more commonly? Why are we using chlorine? Well, it turns out that iodine is a black tar-like kind of chemical. And if we were to try and use it if we were trying to use it in the water would be a grayish color, and people don't like drinking gray water. So that's why they're not using it in water supplies. It actually is just as effective. Now, iodine is most often used as either what we call an iodophore or a tincture. Let's talk about each of these. A tincture is something dissolved in alcohol. Right? Iodine is often seen in a tincture, where it's iodine dissolved in alcohol. The other way that we see iodine commonly is as what's called an iodophore. An iodophore is when you have iodine attached to a polymer, and that polymer slowly releases iodine over time. So you have this slow, extended release of iodine over time. That's what an iodophore is. It's a polymer attached to the iodine that slowly releases it over time. So it kind of increases the length of time that you have effective killing. In fact, you're probably already familiar with an iodophore that's used all the time in medical settings. Betadine. Betadine is an iodophore. Betadine is often applied pre- and post-surgery. And the reason they like it so much is because they'll put that betadine on, and then during surgery, the betadine is constantly releasing new iodine, inhibiting microbes from being able to grow in the tissues. So that's the value of an iodophore. All right, let's talk about our next chemical. Our next chemical here are detergents. Detergents are molecules that act as what's called a surfactant. I'm going to talk about surfactant in just a moment. But detergents generally are polar, meaning they have one side that has a charge to it. Here you can see one side has a positive charge and the other side is hydrophobic. Right? That's what a detergent is. It's a polar molecule. And these detergents act as what are called surfactants. And surfactants actually insert themselves into a membrane and dissolve the membrane. That's what a surfactant means. It's something that rips membranes apart, dissolves lipids. A detergent is an example of this. A particularly useful detergent is called a quat. Quat stands for quaternary ammonia compound. So quaternary ammonia compound, or quat, is an example of a detergent, acts as a surfactant, and it dissolves membranes. So it is effective at killing bacteria and fungi. It's not very good at killing spores. So it's going to kill bacteria and fungi by dissolving their membrane. Now let's look at why it's so effective at killing bacteria. A quat specifically has a positive charge on one side and its hydrophobic chain at the other. What kind of a charge does the outside of bacterial cells have? Bacterial cell walls have a negative charge. So the positive charge here on the quat is going to be attracted to the negative charge on the outside of the bacteria. So just like this. Here you have the quat where the quat's positive charge is going to be attracted to the negative charge of the bacteria, and then it's going to insert its hydrophobic tail down into the membrane. You get enough of those inserting into the membrane, it literally rips the membrane apart. That's a quat. Now, quats are very, very common. We find them in all sorts of household chemicals. In fact, probably it's in several things that you have at your house right now. If you think about all of the different chemicals that you have probably under your sink in order to kill off microbes and to clean, if you were to put the top on it and shake it really hard, if it gets foamy when you shake it, it has a quat in it. Quats are extremely common. You see quats in floor cleaners and mouthwashes, and you see quats in 409, and you see quats in all kinds of stuff. They're very, very common. Actually, after we get through this lecture, I would suggest next time you go to the grocery store, spend a couple minutes just looking at different containers of cleaners and what the active agent is. Most of them will be something that we've talked about in this lecture. Maybe that doesn't sound interesting to you, but it was interesting to me to go through and have a look at what's on those different bottles, and you see how they're killing off microbes. All right, the next one here, phenolics. Now, phenol was really the beginning of the whole idea that we could use microbes or we could use chemicals in order to kill microbes. And what was the name of the guy that used phenol? Lister, Joseph Lister. Remember, all of this activity, all of this using chemicals to kill off microbes starts off with Lister. Lister, who came up with the germ theory of disease, where microbes can cause disease. So Lister used phenol, and I told you about his early experiments where he would force the doctors in his hospital to scrub with phenol before an operation. He would also have the patients scrub down with phenol before an operation. Here's the original phenol. Phenol is also called carbolic acid. It's derived from coal tar. As you can imagine, it worked really well. It's super powerful, but also extremely irritating to human skin. So nobody liked working in Lister's hospital. So phenol here was the original. Phenol works really, really well at killing off microbes. But since then, they have come up with a whole bunch of different what they call phenolics. Phenolics are derivatives of the original phenol. And those phenolics tend to be just as effective as phenol at killing, but much less irritating to human skin. So phenol here is the original. It's very effective, but very irritating. There are a whole bunch of what they call phenolics that are based on that phenol ring structure. They're as effective as phenol, but less irritating. In fact, what's in Listerine is not the original phenol. You wouldn't want coal tar running around in your mouth. It's one of these phenolics that they have in Listerine. All right, so how does phenol and the phenolics kill? They kill by disrupting cell membranes and denaturing proteins. They are bactericidal, virucidal, fungicidal, but they don't get spores. Because spores don't have a membrane that's readily accessible. So phenolics don't get spores, but they do get everything else. Phenolics are quite common. In fact, one of the most common substances to find in like antimicrobial soaps and lotions is called triclosan. It's Triclosan. Triclosan. Triclosan is a phenolic. You find it in all sorts of different antimicrobial lotions and soaps and all those sorts of things. Very, very common. Now, phenol, since it was the original chemical being used to kill off microbes, we often compare phenol to any new chemical. And we do that with what's called the phenol coefficient. For example, let's say that you're a chemist and you have created this new chemical that you want to sell because it's such a great microbe killer, you're going to make $1 million. What you'll need to figure out to put on your packaging is the phenol coefficient. This is something that tells us about how active your chemical is. Here's how you calculate it. The phenol coefficient, I don't know why I have PE there, it should have been PC, but the phenol coefficient is the lowest concentration of your chemical that kills microbes in 10 minutes divided by whatever the results were for phenol. So it's a comparison. The phenol coefficient tells us just how effective your chemical is at killing. And all chemicals, when they're marketed, have to have a published phenol coefficient so that they can be compared to other chemicals. You know how much to use and how effective it is and all those sorts of things. That's all done with this phenol coefficient. All right, the next one here, chlorhexidine. Chlorhexidine is actually a phenolic. Right? So it's one of those phenolics. It kills the way other phenolics do by destroying cell membranes and denaturing proteins. It acts as a surfactant, like you're seeing here, to dissolve the membranes, just like the other phenols and phenolics do. It is a broad microbicide, not sporicidal. Everything here the same as what we saw a moment ago with the other phenolics. So the question is, why does chlorhexidine get his own slide if he's just like the other phenolics? And the answer is because chlorhexidine is exceptionally mild. Chlorhexidine is exceptionally mild on human skin and mucous membranes. In fact, it's so mild that they use chlorhexidine as a neonate wash. They use chlorhexidine to wash out wounds. They use chlorhexidine on mucous membranes or to wash out the eyes. Right? Extremely mild. Effective but mild. So very, very nice that we have this possibility. That's chlorhexidine. All right, next thing I want to talk about here are alcohols. Alcohols are effective killers. Ethyl alcohol and isopropyl alcohol are the two that I'm talking about. The truth of the matter is that methyl alcohol, or methanol, is also a very effective killer, but even fumes from methanol can be very dangerous to humans, so we don't usually use methanol. We'll use ethyl alcohol or isopropyl alcohol. Now, ethyl and isopropyl alcohol in solution somewhere between 50 to 90% are effective killers. They act as surfactants, they dissolve lipids, so they dissolve membranes, and they denature proteins. So for example, if the kid falls down and they've got a skinned knee, you can get out the alcohol, and not only will that alcohol disinfect, it also rubs off the dirt and oil and fat from the surface of the skin. So it's nice with the alcohol, you're getting kind of two things at the same time. Alcohol is bactericidal, it is fungicidal, but it is not sporicidal. It does not kill off spores, but it will get envelope viruses. Envelope viruses have a membrane around them, and alcohol will dissolve membranes. So you can get those envelope viruses using alcohol. The thing you really want to remember about alcohol, however, is that alcohol is the exception to our rule. Normally, higher the concentration, the more effective the killing. But with alcohol, that's not really true. With alcohol, it has to have some water for it to be effective. A 100% solution of ethanol is not as active as a 90% solution of ethanol. Now, the reason I'm driving that point home is because in your future careers as healthcare professionals, right, at some point you're going to have to refill the alcohol bottles. You'll have to refill little squirt bottles that they use to clean equipment. And you better look at that container when you're refilling them and pay attention to if it is a 50% solution, a 75% solution, or a 100% solution. Because if your refill is a 100% solution, you better put some water in those containers. Otherwise, the alcohol will not be effective. Now, many healthcare providers purchase 70% alcohol or 90% alcohol so they don't have to worry about it later, and that's fine. You just need to remember to check the containers because absolutely must have water for alcohol to kill. All right. Hydrogen peroxide. Hydrogen peroxide is deadly because it produces highly reactive oxygen radicals. Talked about oxygen radicals once before when we were talking about anaerobes, and I told you about how living in an oxygenic world, living around oxygen, has its price to pay. In fact, oxygen creates these oxygen radicals that are really quite dangerous and destructive. Aerobes and facultative anaerobes have enzymes to get rid of those oxygen radicals, but anaerobes do not. Well, it turns out that if you have a weak hydrogen peroxide solution, about 3%, then you're going to find that most facultative anaerobes and aerobes can survive because they have the enzymes to break down the hydrogen peroxide, those oxygen radicals. If you use a strong solution, however, like 25%, then absolutely not. They cannot survive in 25%. 25% hydrogen peroxide will kill off any microbe, no matter what. It will destroy anything, even spores. It's so effective. So hydrogen peroxide produces these oxygen radicals that damage protein and DNA. It's a broad microbiocide, and even strong solutions will completely kill everything. They can be sporicidal, so it is an effective way to kill. So for that reason, hydrogen peroxide works really well in strong solutions to sterilize things, to sterilize equipment. You can't put 25% on a human body. You put 3% on the human body, but if you wanted to sterilize equipment, you could use 25%. For example, here, this is an endoscope, and an endoscope is used for colonoscopies. Right? You see the long cord here, and at the very tip of the cord, here is all of the expensive stuff. You see electrodes and video cameras and all sorts of stuff at the very, very tip. And of course, you know where this goes during a colonoscopy. Well, obviously, a doctor's office will probably have multiple colonoscopies that they're needing to run in one day. So the question is, do they need to clean this between patients? Of course they do, right? Of course they have to clean this. Not only do they have to clean it, they have to sterilize it. Well, do you think something like this, with all of this expensive electronic stuff on the end of it, can go through an autoclave? No way. It's going to destroy the equipment. So what they do is, after degerming and cleaning it, you then load this up into this chamber, close the top, and the chamber floods with a vapor of hydrogen peroxide. High enough solution of hydrogen peroxide to completely kill everything that could be in there, and it's sterile before the next patient needs it. So very nice. You don't have to use heat, don't have to damage the equipment. Instead, you're using hydrogen peroxide. It's much faster and less damaging on the equipment. So that's a great example of how hydrogen peroxide can be used. All right, another example here, heavy metals. We have known for quite some time that solutions of various different metals are deadly to microbes. In fact, they're deadly in extremely low concentrations. Solutions of silver and mercury are known to kill vegetative cells in very, very low concentrations. And they do that by inactivating proteins. In fact, if you take this plate and notice that what they've done here is they've taken the agar, melted it, mixed some bacteria into the melted agar, and then poured it on the plate. So they have a bacterial lawn here. See that? Bacteria growing all over the surface. And then what they did is they took a little piece of silver amalgam and they just dropped it right there on the plate. They took a little piece of gold foil and dropped it here on the plate. And the minuscule amount of silver that is leaching from this amalgam into the media is enough to kill all of the microbes around it. That's amazing to think about because there's not very much silver that's leaching out of that amalgam, but the trace levels that are leaching into the medium are deadly to the microbes that are around it. Completely kill them. And the same is true for the gold foil. Drop this little teeny bit of gold there, and the tiny flecks of gold that come off are enough to kill off the microbes around it. So solutions of silver, mercury, gold, all kill vegetative pathogens in very, very low concentrations by inactivating proteins. The bad news is they're also pretty toxic to humans in relatively low doses. So heavy metals, yes, they're effective killers for microbes, but also they're toxic to humans in relatively low doses, so that's a bit of a problem. They are a broad microbiocide, but they won't get spores. They don't kill spores. So usage here is a little bit limited since it is dangerous to humans. There's only some places you can use these heavy metals. For example, sometimes there's these new bandages that I've seen that are for deep wounds, and the bandages actually have silver inside of the bandage. Can only imagine how much they cost, but anyway, you have this bandage and it has the silver actually in it, and you use it to bandage deep wounds, and the idea being, even though there may be pus accumulating in the bandages, no microbes can grow in that bandage because you have all of that silver inside of it. So it's not actually contacting the tissue of the patient, but it's inhibiting infection by keeping microbes from being able to grow in the bandage. That's a great way to use it. All right, the aldehydes. The aldehydes here, glutaraldehyde and formaldehyde, are the two major ones. These aldehydes kill by cross-linking proteins and DNA. Cross-linking means it just attaches two things together. So in the cell, whatever two proteins happen to be next to each other, just because at that point in time they happen to be floating by, the glutaraldehyde or the formaldehyde will permanently attach them together, permanently bind those two proteins together, and that destroys the activity of the proteins, completely deactivates them. You might think to yourself, just how dangerous would it be to cross-link? Well, imagine if we were in lab and I just permanently attached you to whoever your lab partner is, right? It would inhibit your ability to function. And indeed, it inhibits the ability of these proteins to function, and that kills the cell. So aldehydes cross-link to kill off cells. They are broad microbiocides, and they can even kill spores. Particularly glutaraldehyde can kill spores. Formaldehyde we're not as clear about, but certainly glutaraldehyde can kill off spores. Ethylene oxide gas. Ethylene oxide gas is also a cross-linking agent. It will also cross-link, just like we saw a moment ago with the aldehydes, and that inactivates proteins and DNA. They can also destroy spores. Ethylene oxide gas can destroy spores, so it does reach sterilization. Ethylene oxide gas is often used as an option for getting rid of waste products. If you don't go through enough stuff and don't produce enough waste to justify buying an autoclave, one of the things you can do to sterilize waste is use ethylene oxide gas in a chamber. So it looks sort of like a mini autoclave. You put your waste in there, right? And then it will mix the ethylene oxide gas and flood the chamber with the ethylene oxide gas, and that will sterilize. It will kill off anything that's inside of there. And then it's safe to throw that trash in the regular trash can. So some offices use ethylene oxide gas instead of an autoclave in order to sterilize. Ethylene oxide gas is also used in food prep and drug prep when you can't use heat or steam heat like an autoclave. You could use ethylene oxide gas to achieve sterilization. For example, many spices are packaged with ethylene oxide gas. If you think about something like your jar of cinnamon, you couldn't send that jar of cinnamon through an autoclave. If you did, you'd get out a little blob of goo in the bottom of your jar. You wouldn't have cinnamon any longer. So what they do is after they fill the cinnamon jar, they pump ethylene oxide gas into the jar and then seal it closed. The ethylene oxide gas isn't going to make it muddy and liquidy, but it's going to kill off anything that's inside of there. You buy that jar, pop off the seal, and the ethylene oxide gas dissipates instantly, and it's safe to use. Right? That's a great example of a way to use ethylene oxide gas to sterilize without having to worry about destroying the integrity of whatever that substance is. All right, I'll see you guys with the next lecture, which is chemotherapy, the drugs.